Okay, Mikey.
We good?
Yep.
Okay. So this is basically fake setup of everything.
Guys.
So we'll have these plates ready, which is the bottom auger, the minimal auger plates. It looks like nothing's on here, but there's a thin layer minimal auger. So you'll have those all ready and labeled. Then to start off with, we'll start in this area is where you start. You have your tube. You made the liquid culture overnight. It was in the incubator. You take it out of the incubator when you're ready to use it the next day. Then bring it over to your bench and whatnot. Then you pipette some of the cells into a different tube. Into the different tube also goes the mutagen, if you are using mutagen for that control or experiment. So from here, you're going to incubate that for the 20 to 30 minutes. During the 20 to 30 minute incubation, so this is in the incubator, right? During that incubation, you're going to heat this to liquid. This is the top auger. See how it's like jelly but like solid? So you can't quite pipette it out yet. So just microwave. Then once that's microwaved, and this is done in the incubator from the 20 to 30 minutes.
And what's in that tube again?
In this tube is the cells and the mutagen. Sorry, I'll go over here so everyone can see. The cells and the mutagen are in this tube. If you are using the mutagen, the formaldehyde, remember you take out the 150 microliters and transfer it in order to get the correct amount of cells, just into a different one of the same type of tube. And then you add 2 milliliters of this, which will be in a liquid form, to the tube with the cells and mutagen in it. So you did this step, microwaved it, heat it, so now it's a liquid. You add that into the cells and mutagen that was incubated for the 20 to 30 minutes. You take that. These tubes are a little bit different because we don't have the plastic ones because somebody ordered improperly. But you just take them, my hand fits on it, see? Just flip it once. This is the step that needs to be done really quickly when you're pipetting the 2 milliliters of the top auger into here and inverting to mix and pouring, because the top auger will harden. We went over this. So you have right here, and then you just take the plate, open it, and just take the top off. You can put it in the trash, just pour it on, and then to mix it, I just like move it around on the table like that.
Can you move so we can?
Yeah, sorry. Just like move it. I don't have anything in here, obviously, but like just move it around. It hardens pretty quickly, so it'll be like sort of jelly form.
So in that plate, there would already be bottom auger, right? Which would be the minimal auger. She would be adding the top auger to that plate, and then swirling the top auger, which would already have the mutagen and the cells in it.
Yeah, it'll have the cells, the mutagen, and the top auger. That will make this liquid harden, basically. And we'll also have the tryptophan in it or not have the tryptophan.
Don't forget microwave before cells. Don't microwave the cells.
Microwave this bottle, not the tube. Then when you put this back in the cabinet, it'll just re-jello form. Then with these plates, you'll have a lot of them, obviously. Invert it, put it in the incubator 37 degrees overnight, or room temperature for 2 weeks. Or 3 if it's okay. Questions?
Somebody's got questions.
Somebody has to ask a question right now.
I will expose you as a question haver if you do not ask questions on your own. Compost, I can tell you have a question. No? You understand this protocol? So if I ask you a question about it right now, you'd be able to answer it? Gabby? Gabby? Jackson? No questions. Arjun, new guy? Questions?
No, I think I understand.
Okay. Then why don't we go over the different experiments?
Yeah, okay, so the different experiments. So we'll just do 1, 2, 3, 4 as the controls that are on the paper. And then 5 as the actual experiment if you're testing a different chemical other than formaldehyde. Does that make sense to everyone?
Why don't we just go over quickly the point of this whole thing?
What do you mean?
What is the AMES test designed to do?
The AMES test, you'll be able to tell if some sort of chemical causes a mutation to the cells, meaning that it can potentially be carcinogenic because it was causing that mutation to the cells.
How are you going to do that?
You do that by the cell in this number 5. The cells are not given any tryptophan. So what does that mean? Okay, it won't have food, right? And it won't be able to grow. But if that chemical causes the base pair mutation, like the formaldehyde does as a positive control, that means that chemical is able to cause the mutation, correct? Being mutagenic, being possibly carcinogenic. Good.
But this test only proves that the chemical is mutagenic, right? Not carcinogenic.
Can you speak louder, Liz, please?
This test only proves a chemical will be mutagenic, right? Not carcinogenic.
Yeah, it's one of the first tests. Let's say a new chemical is invented, it's one of the first tests, and it's a broader indication of whether it's carcinogenic. Okay. Does that make sense?
Yeah.
Questions? Okay, so we can go through all of these. And then, so where do you put the mutagen in this process?
Do you mean like which one too?
Yeah, like is it in the top auger or is it in this mixture?
One and four.
Gotcha, Kim. You're fine.
Sorry, what are you? What are you
was the
asking?
question?
Where?
Okay, in this process, where is the mutagen?
Oh, it'd be in down. Yeah.
Here, right? Okay.
Yeah.
So then, just so we know where everything is. So we know it's with the cells in this incubation process. Top auger will have
Tryptophan plus or tryptophan minus.
Plus or minus tryptophan, and the bottom auger for all of these will be okay. So for number 1, it will have tryptophan, so the top auger will be positive with tryptophan.
Do T plus.
Okay. They look the same though.
It's capital T. Okay.
T plus. Okay. It will have. I don't like that.
So do bottom auger is T minus.
Yeah. Bottom auger won't have any tryptophan. And the mutagen, it has a control chemical, meaning positive.
Can we call it positive mutagen?
Yeah, sorry.
And the mutagen, the positive mutagen, or control mutagen is what?
Formaldehyde.
Very good.
Okay. Makes sense to everyone.
What would you expect to see here?
Yes.
Okay.
Did you expect more growth than normal here, or is that not
I would expect less.
Um, What?
He's asking if you would expect.
Yeah, when I did it, it was, 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 it was it was pretty much a long growth, so you can't really tell that well, just because it had the tryptophan and the formaldehyde was also mutating the cells, so it just looked like a ton of cells all over the plate.
Okay.
Yeah. Okay, control number 2. So it has tryptophan, so it's T positive in the top auger. It doesn't have a control chemical, so minus mutagen. And you also are still using the minimal auger plates, so what will that be? Questions?
What is number 1 testing for?
Just growth of the cells. It's like
Number 1 is
If you were testing for growth in general, then, well, okay, so what's the difference between 1 and 2? What is the difference between 1 and 2 are testing for?
Whether or not the mutagen affects the growth.
Whether or not what?
Whether or not having the mutagen affects the growth when there's tryptophan.
Exactly. So did everybody get that? In 1, we'll know if the mutagen is killing off all the cells. Because when we compare 1 to 2, the only difference there is the mutagen.
Yeah. That goes into a little bit of the cytotoxicity levels. So if the formaldehyde concentration is too high, it'll kill all of the cells, regardless of having tryptophan or not. Make sense? Okay. So what would you expect it to? Okay. Number 3. Somebody else fill it out. Jackson?
minus.
Yeah. Minus mutagen. Because it doesn't have a control chemical. It doesn't have tryptophan, so the top auger is T minus. And the minimal auger plates that we're still using are also T minus. What would we expect to happen? Okay, and the
What does 3 tell us?
that without the mutagen and without any food, cell should be able to grow.
Without the mutagen,
Without the ability to mutate and without any cell food, there should be no growth.
there should be no growth. So what are we trying to distinguish here?
We're trying to see
that makes a difference in the growth of those cells.
What if we saw growth in number 3?
You saw growth in number 3
Then
that
they're mutated.
tryptophan doesn't play
They're
a role.
mutated cells.
The cells would already be mutated, right? So what kind of control is this? A positive or a negative control?
Negative.
It's a negative control, correct?
Okay, questions? If that grows, it could be contaminated as well. Just another side comment. Okay, so number 4 is the positive control. So there will be a mutagen, so positive mutagen.
It's
There will be no tryptophan in the top auger, T minus, and we're still using the minimal auger plate, so what will that be? T minus. What do we expect for this plate?
mutated cell.
Yeah.
And what does that look like, Mikey? Is that lawn?
No, it's individual colonies. Just because cytotoxicity is
Okay, so in 1, 2, 3, and 4, which ones have selection? 4, 3.
What do you mean by selection?
Which experiments are going to select cells? 4 and 3. Right? Because the tryptophan minus is going to select.
Right.
Now, which ones are going to mutate?
1 and
1.
Mutagen positive.
Yeah.
Why is 2 not selected?
Because it's T plus. Selected ones when you have. And there's no mutagen.
It's just everything.
Or when everything grows.
All cells can grow.
Okay, and then what would 5 be?
A different mutagen.
So we haven't actually done an experiment yet.
It's your fault you didn't order that one.
Shut up.
So then what did you do? 5, 6, 7, 8, 9, 10. Like, don't you have to do with whatever thing you do, what you want to do it with all, or no? You don't have to.
Can you explain what you're saying again, Pink,
I
and
think
louder,
I
please?
understand, but ask again.
If you, for the experiment with the new mutation, you have to redo each control with the new mutation, or can you just do?
No, because you use formaldehyde as that control because we know it's a mutagen. Yeah, does that make sense?
Yeah.
So you're using a different mutagen to compare to the ones you have to see if it's
Correct.
effective
Yeah,
as
you
formaldehyde.
know formaldehyde causes the mutation. The question is, will this other chemical also cause that mutation? But for the controls, we want to know that it is causing the mutation.
I'm saying when you do like one with tryptophan and one without tryptophan, or it's just
You can. It's not necessary to though, because you just want to know if it's mutating or not. You won't be able to tell on the plate that has the tryptophan because it'll just be a long growth of all the cells.
What if your mutation? What if your mutagen has tryptophan in it? So Liz is saying that there should be 5 and 6. It should be experimental mutagen, right? And then it should be a T minus plate, T minus top, and a T minus bottom, and then a experimental mutagen, T plus top, and T minus bottom. So that would mean basically the minimum number of experiments you would have to run at any given group would be 1, 2, 3, 4, double the experiments, 5, 6, 7.
It's because the other control would be considered, the other chemical with the tryptophan would also be considered an experiment because you don't know if it grows or not, so you would have to do 2 of those.
Right.
So it would be 8.
So I would run at least two 5s and at least 2 6s. I would never run a single 5.
Right.
Yeah.
I would never run a single experimental group.
Yeah. But saying you'd have at least 8 different categories.
You'd have 6 different categories, 8 different experiments.
Yeah, okay, that's what I meant. Yeah.
What would happen to you if you ran 1 5 or didn't run your controls? Jackson? What's that? I would kill you, correct. Very good answer. Very good answer. So, yeah. So down at the bottom where it says growth, growth, no growth, muted cell, it'd just be like, I would die.
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 cytotoxicity of all human cells.
5. You have to have
Okay. Guys, I think we owe a thank you to Mikey and her mentor for this project, Dr. DeCilio, who worked on this for an entire school year. So let's give them a round of applause. Mikey, bow or curtsy?
No.
Mikey?
I really want to do something, but I won't do it on camera.